Yeah.
A very good morning, one and all. I, Dr. Jagruti Desai, extend a warm welcome to all the participants, guests, and my dear friends to Charuset Endowment Chair on Regulatory Considerations for Pharmaceutical Development, our global scenario. Organized by Ramanbhai Patel College of Pharmacy and T.P. Patel Center for Postgraduate Studies in Pharmacy, Charotar University of Science and Technology. I now request our principal, Dr. Manan Rawal, to take the function ahead with his address.
Thank you, Jagruti, madam. Today's speaker, Dr. Ranjit sir, the audience, staff members who are present over here, faculty members, ladies and gentlemen. It is my real pleasure and honor to say a few words about our university and the institute. Our university, Charotar University of Science and Technology, has been conceived by Shri Charotar Moti Satavis Patidar Keravni Mandal. This is not-for-profit Premier Education Trust of India, and we have a social lineage of more than 119 years. Charuset has been established under Gujarat Private University Act number 8 of 2009, and Charuset now has been awarded A grade by NAC. Recently, we have considered for a Center of Excellence. Yesterday only it was announced, and now Charuset has been one of the Center of Excellence and awarded by Government of Gujarat. Presently at Charuset, we offer more than 52 undergraduate, postgraduate, and PhD programs under the total age of 9 institutes and 6 faculties, including 5 centers. They are in allied disciplines of technology and engineering, pharmacy, computer applications, management, applied sciences, nursing, physiotherapy, and other paramedical sciences. So our institute, Ramanbhai Patel College of Pharmacy, RPCP, is a constituent institute of Charotar University of Science and Technology. The institute was established in year 2004 with the patronage from Shri Pankajbhai Patel, Chairman and Managing Director of Zydus Cadila Healthcare Limited. At RPCP, we offer 5 MPharm programs, namely Pharmaceutical Technology, Regulatory Affairs, Quality Assurance, Pharmacology, and Pharmacy Practice. We do offer a few certificate programs in impurity profiling, IPR, and there are several areas we are exploring. We do offer PhD program in all the areas of pharmaceutical sciences. With this brief, I again welcome you all, and I sparing a time, and that is huge time, like 4. That is really great, and sir, we are exciting to listen the QBD approach from a person who has decided and has contributed in devising many of the guidelines where QBD is actually applied. So with this, audience about this function, please. Thank you.
Thank you, sir. I request the coordinator of this program, Dr. Gayatri Patel, to introduce the participants with our event.
Thank you, Jagruti madam. Good morning to everyone, respected Ranjit sir and dear participants. I, Dr. Gayatri Patel, Professor, Department of Regulatory Affairs, RPCP, and coordinator of this event, feels an immense honor and pleasure to welcome all the participants to the present event. I am extremely glad to introduce our event, Charuset Endo Chair, which is an endowment fund created by Charotar University of Science and Technology. It is established with a purpose to promote academic activities as well as to promote excellence in research and innovation. At RPCP, we organize this endowment chair activity every year with an objective to provide the platform to our students for interactions with the eminent speakers, scientists, and professionals of various organizations. Usually, the invited expert stays at Institute for 3 to 4 days, deliver their talk, and interact with faculty members, and sometimes they solve their difficulties also. Dr. Samin Mitra Gotri, Dr. Vanna Patrawale, Dr. S.Y. Gabesar, and Dr. M.C. Gohil were invited in the past years. This year, the pandemic situation has enforced us to arrange the endowment chair activity through online mode, and we consider this is instead as an opportunity. We extended the benefit of the rich technical sessions to the students of other pharma institutions by streaming the sessions to the live on YouTube along with the platform subscribed from Zoom by the institute. This is not going to without any financial implications to the students, and all the participations will be of free is a free of charge. And after attending all the sessions, they will receive the certificate for participations. With a great pleasure, we announce Charusat Endowment Chair activity at RPCP year 2020-21. The theme is regulatory considerations for pharmaceutical development, a global scenario. The consultant and experts from the consultancy firm and pharmaceutical industries is going to deliver the sessions as a part of this activity. This activity is spread over for 5 days, 1st June to 5th June. And the details of the session is already delivered to all the participants. But here just I give the glimpse of the expert, Dr. Ranjit Barsikar, sir, he is a CEO, QBD International, and member of the Journal of Generic Medicines, UK. Then Dr. Hardik Soni, sir, Senior Manager, R&D, Vasu Research Center. Miss Bijal Shah, Assistant Manager, R&D, Vasu Research Center. Miss Meena K. Shah, Pharma Quality and Regulatory Advisor. She is working at the Tanvi Associates, Mumbai. Dr. Rajashree Ojas, ma'am, Founder and Director, Raj GPRAC Private Limited, Mumbai, India, and Dr. Jignesh Patel. These are our eminent speakers who will deliver the all the subsequent sessions or the topics they have given us. With this, I welcome all the participants once again. Now, Jagruti ma'am, it is over to you.
Thank you very much, ma'am.
Thank
you.
So,
moving ahead for today's session, we have with us Ranjit Barshikar, the CEO of QBD International United Nations Advisor, Geneva Editorial Board Manager, Journal of Generic Medicines, UK. In his career of around 51 years, Sir has a rich and varied experience in the pharma industry, especially in the quality management, R&D, manufacturing, US FDA, European Union, WHO, etc. compliances. He is also an expert on quality by design, data integrity, CAPA, quality culture, and various other CGMP subjects. He is also an expert auditor of formulations and API facilities, CRO, etc. He has worked as quality expert with World Bank, Washington, USA. He worked with Saudi Arabia FDA for generating the regulations and training SFDA inspectors on behalf of World Bank. He has also worked with Pfizer Limited, Sun Pharma, Leica Labs, Cancer Research Institute, Mumbai. His major awards includes, he received the Quality Champion Gold Topmost Quality Award by the Government of India, Quality Council of India. He also received International UNICEF Council USA Award and International Lifetime Achievement Award. He has also received 100 Most Impactful Healthcare Leaders Global Award by World Health Congress. He has also received the Pharma Ratna Award by largest NGO, Rabdi Meher, and in addition to that, he has received more than 35 other awards also. So I invite you, sir. We have pleasure to have you here for this first session. And today he's going to speak on QBD regulatory guidelines, ICH, US FDA. So now I request sir for the session.
Thank you very much, madam. I appreciate. Thank you very much, Principal Manan sir, Dr. Gayatri, for inviting me for today's session. It has been always my pleasure and passion to support the universities and the pharmacy colleges globally. And this is my 38th webinar in last 10 months, which includes various conferences as well as the university talks on various pharma subjects. So it's the whole concept of this 5 days program with the theme of regulatory considerations for pharmaceutical development, a global scenario, this theme is very interesting, and I really appreciate. I haven't come across this kind of theme in any other program as well. There are different nomenclature, there are different headings, titles, but this regulatory aspect, which is most crucial in addition to other part, is missed by some of the institutes, so which you have captured. So I appreciate it very much. And my talk today, there are two talks, one in the morning and another in the afternoon, 3 o'clock. Morning, I will be speaking now on regulatory guidelines, ICH and USFDA on quality by design. And in the afternoon, I will be speaking on quality by design, product development, current scenario, and future challenges for the product development. With your permission, I'm now sharing my presentation and then start my presentation. Okay, and if you have any difficulty, please inform me in between. Okay, madam?
Sir, there is a little problem here. The audience cannot directly be able to interact with you. So we have question answer session at the end.
No problem.
So we have kept some time, 15-20 minutes somehow for question answer sessions.
I understand, but you have to, you please able the screen sharing for me. It's not happening.
That I'll check it out.
Yeah. The message I get is host disabled screen sharing.
Yes, sir. Just what.
Now I'm sharing.
Okay.
Are you able to see?
Yes, sir. It's fine. Perfectly visible, sir.
The full screen?
Yes, sir. No, sir. You are not on full screen.
Okay, I'm just then disconnecting and then again joining. Well, I'm resharing it again. Hold on. Now?
Yes, sir. It's fine. It's completely full in full mode. Full
Okay.
screen mode.
So, friends, let's move ahead with our subject today on the quality by design regulatory guidelines, keeping in mind mainly ICH and US FDA expectations, their guidelines, and which is a key thing for all of us to know in details. You may be having some of this information available in your syllabus, but it's always good to know how industry perceives it, how industry looks at it about these kind of regulatory guidelines, and how industry is expecting it and how it is being implemented in documentation as well. So from that perspective, I really appreciate from this regulatory guideline subject to the students. I'm sure you must be aware of these quality guidelines which are there available in public domain. If you visit ICH, International Conference of Harmonization, and this is a body which is, there are 3 countries party to this. One is America, another is Europe, and third is the Japan. And these 3 together have come to form ICH board, and who have developed these guidelines, and these countries are implementing it for their expectation for world to export to their countries. So we are going to focus on these guidelines, ICH Q8R2, that is a pharmaceutical development. It's a revision 2. R2 is revision 2. Q9 is quality risk management guideline. Q10 is pharmaceutical quality system. Now, when we talk of the product development, we are referring here formulation development. And these three documents we have to understand, read and understand in considering three together. And then same concept is also applied to ICHQ11, which is a development and manufacturing of drug substance, that is API. And now recently finalized guideline in ICHQ12, which is called life cycle management. And this life cycle management of the product, whatever time the product is in the market, by the organization, they have to monitor the process, they have to improve the process till the product is existing in the market. That is called life cycle management. And ICH is working on two more guidelines, that is Q13, that is for continuous manufacturing, and Q14 is for analytical method development, validation, especially keeping in mind. So these last two, 13 and 14, are yet to be finalized, but the work is on. Whereas up to Q12, all are final and people are already using these guidelines. So all these guidelines of ICH are also adopted by US FDA and European agencies as well. Look at here this document. Now, when this was available, this Q8R2, that's a pharmaceutical product development. And if you see here, it is available since more than 12 years, more than a decade. And this is in use. Before this, the first document, which is only Q8, was available in 2005. And further it is revised as R2 and since November 2009, revision 2. Remember this, revision 2. So this is how the ICH and US FDA are viewing these guidelines. Q8R2, Q7A for API GMP, Q9 for quality risk management, Q10 for pharmaceutical quality system, and Q11 for the life cycle management Q11 is for API product development, and Q12 for life cycle management. So why these were developed? These were mainly developed because, keeping in mind the whole concept of quality by design is science and risk based, and this is a big paradigm change in the compliance perspective to ensure the product is robust. So whole concept of science and risk based approach in the development of the product, which encourages step by step designing the quality into the product, not only the end testing. This I'm going to discuss in the evening more, which is applicable to the product life cycle as mentioned in ICH Q12 guideline, and to enhance the quality of the pharmaceutical product in the interest of the customers, that is patients. Now let's look at what Q8R2 says. ICH Q8R2. It says science and risk-based approach for the product and manufacturing process development. And here it is manufacturing process development with the help of the main concept behind quality by design is the design space. Why design space? Because when we are having a particular normal range of operations for critical quality attributes or critical process parameters, you have to establish that your product complies fully with those ranges with the help of design of experiments, which is a part of design space. So design of experiments are supporting to define design space. And there we have a regulatory approach of flexibility of the to play within the range. And this quality by design concept and some examples given by US FDA for the development and how you can develop the product and how you can develop the design space with the help of design of experiments. So what it comprises of? The product development through quality by design as mentioned by ICH Q8R2. It is quality target product profile. That is the first step. And this based on the customers. Identify the potential critical quality attributes, linking the input material, that is raw material attributes, input material, which also includes the excipients and API together for identifying the impact of critical process parameter on CQAs with the help of risk assessment. Develop design space. Though it is not mandatory, design space is not mandatory. It is optional. But they do ask a lot of data which makes you to develop a design space. And based on the design space, how do you monitor all the critical process parameter during manufacturing? And how do you continuously improve the product throughout the life cycle is a whole concept of QBD development. If you look at the ICH Q9 guideline, it's a quality risk management, which is the key of whole activity and quality by design concept. It describes the systematic processes for risk assessment to control, to communicate and review the quality risk during manufacturing. And this is the way you are designing quality into the product. That is very, very important to keep in mind, that how we can develop a product, what kind of quality risks and how to identify these, how to identify critical quality attributes, how to identify critical quality parameters, and how to monitor it. There is nothing beyond this. But the whole thing is the risk management. How do you handle? How do you understand it correctly? And that's the way to improve your knowledge of the product, to improve the understanding of the process and the product as well. And that is why we say that assessment of the risk to the quality should be based on scientific knowledge, linking to the safety of patient and throughout the life cycle of the product. This is how this whole process of quality risk management works. If you see this, the diagram on your screen, it is mainly in three parts. The whole process of risk management is in three parts. First is assess the risk. Second is plan to control the risk. And third is review the risk, communicate and go on improvement as you gain the experiments of your process. So what happens in the risk assessment? When you do the risk assessment, in that you have to identify where my process can fail, where my manufacturing process can fail, where my equipment is likely to fail, where my testing procedure is likely to fail. So identify it first, that what can go wrong. Then analyze that risk. After identification, analyze the risk. Once you analyze the risk, then evaluate the impact of the risk on the quality. The impact of the risk on the quality parameters, on the quality attributes. Once you know it, then you can plan to control, to monitor, to generate the monitoring process. What is monitoring process? You are generating procedures to monitor the process, to control the process so that it should not fail. That particular critical process parameter should not fail. And that's the way you are saying that I am accepting the risk and I have monitored the risk also, that now it will not fail my process. And then we are going ahead to review the risk and communicate completely throughout the life cycle with the help of various risk management tools. So this is in nutshell the whole activity of quality risk management. Look at this. So first part is process development for the risk perspective. Second part is control strategy during the development, that is risk control. And the third is improvement throughout the life cycle. Remember these three steps in the risk management. Next guideline of ICH is, which is to be read along with the product development quality by design, is pharmaceutical quality system. It describes the key systems that facilitates establishment and maintenance of the state of control. So whole concept, or how do you ensure the product compliance in the interest of patient is looking from the perspective of the state of control. How do you control the whole process for the critical process parameters, and how these parameters will perform to enhance the product quality, which facilitates the continuous improvement with the help of the Q8R2 guideline and Q10 together, with a strong support from risk guideline, that is quality risk management Q9. So all these three together are supporting us to track and train the quality, maintain and update as and when required, and continuously verify the process changes are successful or not by way of process validation. And this verification comes at the third stage of process validation, that is called continued process verification, CPV in short. So this diagram will brief you in nutshell what we have been talking, that the fit for purpose of the quality system is as our knowledge of the product and process increases, look here, increases, I'm repeating again, as our knowledge of the product and process increases, we are reducing the risk of the product failure. That's the key to handle the whole development process. And where right from the initial development till commercial manufacturing, we have to handle whole this process in the keeping in mind the patient safety and their expectation that you will have a consistency in manufacturing. And in this consistency, ensure not a single process step will fail, and you are successfully monitoring by identifying the right risk involved by right control strategy. So keep in mind throughout the commercial manufacture and lifelong of the product, we have to monitor this. So what is the key here? As your knowledge of the product and process grows, the risk of the product failure goes down, remember. So if you see this diagram now on the screen, this is what exactly we are referring as a part of quality system, ICH Q10, and this is applicable throughout the product life cycle. Remember this, pharmaceutical quality system is a process itself, and we have to integrate quality by design whole concept into the quality system. And right from the product development stage, technology transfer, commercial manufacturing, and whenever it gets discontinued, maybe after 10 years, maybe after 50 years, maybe after 100 years. So throughout, we have to monitor this activity. And this has a complete linkage integration with the good manufacturing practices, guidelines, which are separate guidelines. As per US FDA, those are 210, CFR 210 and CFR 211, where you see as an enabler for quality system implementation. Look at the foundation here. Look at this. Quality risk management and the knowledge management are the key strong foundation on which you are building your pharmaceutical quality system, into which quality by design is integrated. And that is the key. And this foundation must be very strong. You have to be highly knowledgeable. You have to understand your processes. You have to understand your product, and you have to use the risk management understanding to develop and integrate quality by design in the elements of the quality system. What are those elements? First is, of course, process performance and the quality monitoring system. Then CAPA is corrective action and preventive action, that is called CAPA system. Then change management system. And finally, the quality team should report to the highest management in the organization, and they're reviewing every quarter or once in 6 months whole performance of the quality based on the product performance. And this will strongly integrate with the pharmaceutical quality system of quality by design. So the key here is quality risk management and knowledge management with the help of management responsibilities of various activities, as I mentioned, CAPA, change management, and process and product performance, combining with the good manufacturing practices, have a commercial manufacturing, throughout the life cycle of a product. So it is very, very important. Let's go to the another ICH guideline, life cycle management. Since I was just talking to you, I thought to jump over quality, the Q12, and then afterwards come to Q11. Here, what happens is whole Q10, the quality by design integration into the pharmaceutical quality system, gets integrated by way of fit for purpose understanding. What is fit for purpose? Is safe, efficacious, and quality product for the customers, for the patients. So where we have to understand and consider raw material qualifications, handling the changes and deviations, calibration, qualification, validations, manufacturing records, documentations from that perspective, analytical method development and implementation, and the specification. All these are part of life cycle management, and here we can include also the stability data of the product in the life cycle management. So when we combine Q8, Q9 and Q10 from the development perspective of a product formulation, we have to keep in mind, one is formulation activity, another is process development activity, and third is manufacturing perspective. These are the three parts where these three guidelines have to be used and understand for implementation together. And ICH Q11 was subsequently added. Initially, these three were there, Q8, Q9, Q10, and Q11 API was subsequently added, which is supporting the Q8 strongly. Let's see now how these ICH Q8, 9, and 10 support to the industry, how it can be helpful, and how it is so valuable for us. And we appreciate the regulatory agencies for developing such guidelines. And if you see, what it combines is the drug product, the development approach, and the regulatory system together now. From the innovator perspective, that is a patent holder perspective. And when we manufacture as a generic, it is a generic perspective, which I'm going to discuss more in the evening, my session. The small molecules are the chemical molecules, pharmaceutical products, whereas biotech are a different molecules, which are large molecule beyond, say, more than 30, 35 amino acids is a peptide, and beyond 50 amino acids together, it makes a biosimilar product. And there are several biotech which also include blood products, which also includes protein, antigens, antibodies, etc. And nowadays we are having a hot subject in discussion world over on vaccines. So it generates a lot of antibodies in the body, a lot of proteins, the malfunctioning proteins are attacked. These antibodies are playing key role into that. So we are moving away from traditional development to quality by design with the help of these guidelines. And it's a key to perform so that your product will never fail, and you will have a consistency in batch to batch manufacturing, unit to unit in manufacturing, and every day throughout the life cycle of a product. So as I mentioned to you earlier, I'm just briefing you here the scope of these guidelines. In short, is process validation guidelines, quality system, and the pharmaceutical quality system. Q10, Q9, pharmaceutical analytical technology guidance FDA has, ICH Q8, right throughout the product design, to the process monitoring control strategy for continuous process verification throughout the life cycle of a product. Whole activity with the help of good manufacturing practices all work in tandem to give a satisfaction to the customer for the product they get. There is another guideline which was issued by FDA in 2002, almost 2 decades ago, and that is called pharmaceutical CGMP for 21st century. That is risk-based approach, which the objectives were implementation of QMS or quality management techniques, implementation of risk-based approach, and state-of-the-art pharmaceutical science, and which resulted into the current guidelines and the US FDA regulation for compliance perspective. So how these are all mixed together, and for implementation, how they have hand in hand to each other for the process development and manufacturing improvement throughout the life cycle. It is science-based, optimizing your process from the designing the product and then going for technology transfer for CPPs and CQAs, and at the site, manufacturing site, implement it same thing and ensure all your facilities are working okay and qualified, and then go for process validation completely. All these guidelines, Q8, Q9, Q10 are together with the help of good manufacturing practices in manufacturing, in monitoring the critical processes with the help of right analytical techniques or tool to have a successful implementation of the guidelines. Let us look at this. I have picked up this. I had attended few programs of US FDA, and where they are talking on very interesting aspects that when you implement this quality by design, what is expected by regulatory agencies. They say that we are looking for critical quality attributes in the finished product. What are those? How do you justify this? So when you present this data to the regulatory agencies, we have to justify to them that why you decided out of 30 quality attributes or 40 quality attributes, 15 are CQAs. Why? You have to justify it. When we justify it, you have to put it in a particular tabular form. Some of the parameters I have picked up here as quality attribute and from the product based on the risk assessment. When we write the parameter of the quality attribute, what is the target of that? Say, for example, identification of what? Drug substance. So is it critical? If you say yes, then you have to write it is why. If content uniformity in tablet, then write which procedures you are going to implement. And if it is critical, yes, then why. You have to give this kind of simple tabular form to regulatory agencies. So what beyond that they look for means the CQAs against the input material during manufacturing, their compatibility, the impact on the CQAs of the product and the material attributes, excipient attributes in this kind of form. For example, we say the sodium lauryl sulfate. This particular last dissolution column says high red color, red, high risk. isn't it? When it says high risk, then we have to ensure or develop a control procedure in such a way that this high risk is monitored during the manufacturing activity, so that dissolution will always comply and patient will not have any problem. Same concept for every other parameters which are listed here from the use of the excipients in this tabular form. QTPP and CQAs, the same thing, quality target profile and critical quality attributes are not listed properly in the documentation. You have not understood the process because your risk assessment tools are not fully used by you. You had not given any justification of risk assessment. Some high risk variables are not addressed, are not considered by the company. Look at these, the issues identified by the regulatory agencies. In the designing and understanding the product and process, they say some examples of the deficiency that if the product is bilayer tablet, provide us information that you have the adhesion properties of the two layers is proper, and you have done risk assessment based on that. The double layer tablet, there is no incompatibility issue. They would like to have these documents from the company. So these one layer and the second layer on the top of that, we have to justify, which was not given to them by some organization. So they have reported these as a missing thing. Another is during roller compaction of the tablet manufacturing, visual observation, that is roller speed and auger specifications during compaction are studied. So provide us the data to demonstrate that the allowed variation in ribbon density, that is compaction, will not negatively impact the dissolution. In addition, the roller pressure can have a risk process parameter. So discuss this impact as well. So if you see from these kind of simple examples, how the few companies, when they submitted documents, missed certain things for submission. There is another guideline which I was telling you earlier, is the drug substance ICHQ 11. The purpose is to develop and understand the manufacturing process of API drug and give the clarification with the help of the 8, 9 and 10 guideline. Now, this Q11 is supporting strongly the Q8 as well, Q8 R2. And this, now it is suggested that to identify the CQAs, critical quality attributes of a finished product, take the help of drug substance CQA. That is quite helpful, and this document has a various chapters or the titles on scope, manufacturing process development, manufacturing process and the controls, how you can select the starting material and sourcing the material suppliers, the control strategy of variables, the process validation, how you will file the documentation in the form of CTD. CTD is technical document to be submitted to the regulatory agency, common technical document, CTD. Throughout life cycle management, and some examples are there. This guideline which I was talking to you, Q12, life cycle management of the product, and it was draft was revised and it was given in 2017. And this says that we are supporting through these guidelines for the innovation for continuous improvement of the product. and the continuous verification during manufacturing throughout the life cycle of a product. As I mentioned to you earlier, life cycle of a product is till the product is commercially available in the market by a particular company. It may be 10 years, it may be 50 years, it may be 100 years. That is the life cycle of a product for that company. And risk-based regulatory oversight and harmonize the change management. Whole activity is part of the life cycle management, and where you can always take the help of the stability profile of the product. And it mentions established conditions. This document ICH Q10 says, Q12 says established conditions. That is specifications, how you establish with the help of the design space. And how do you handle post-approval change management protocol for the life cycle of the product, for the quality system, the risk regulatory documentation submission, the inspection of the product, the inspection during the visit to the site, and various references and etc. The lot of data is available when you refer these guidelines. So overall from Q8 to Q12 and how do you see this whole regulatory tool is right from risk-based strategy to the post-approval changes throughout the life cycle. With the help of Q8 to Q11 guidelines, we are strongly supporting the Q12 expectation by regulatory agencies, and which is the key. So its objective is product realization, continuously monitoring the state of control and ensuring your monitoring process is perfect for every batch, every month, every year throughout the life cycle for the improvement of the product. And what is established condition, it says, is description of the product, process, facilities, and elements associated with control strategy. So each control strategy, you have to establish the specifications, the range for implementation, the normal operating range, and acceptable, the pre-approved range, PAR. And these are the different elements which I have discussed with you already, from the established conditions to the post-approval changes. And the key area is annual product review, which is a part of the life cycle management. Annual product review, whole year, how your product has behaved. Are there any issues? They need the summary to be given to the regulatory agencies when they visit the site. And the scope is API and pharmaceutical biotech products, the combination product, the device as well as the drugs, and the changes needed for that. And what they are saying constantly as a part of life cycle management is continuously improve the product with the help of ICH Q12 guideline to reduce the variability of your input material, whether it is excipients, whether it is active drug substance, and whether it is packaging material direct in contact with the product. All these you have to understand the various sources of variabilities. All the sources of variables, address those if it is impacting the final product quality. And the role of quality assurance here is major for established conditions establishment for the critical quality attributes and critical quality process parameters. because we are referring to the established condition, that is a specification for critical quality attributes, critical process parameters. US FDA has released a document. This is also in public domain. It is available at US FDA site. If you are not able to get, let me know. I will share that document with you. I have that the copy of that link. You can always access from that link to get this document. And it is known as quality by design for ANDA submission. This is for generic manufacturers. The more generic manufacturer, what it is, everything I'll discuss in the afternoon. And this example is for modified release dosage form. The complete detail is there for almost 200 pages, stage by stage, how the product is developed. There is another document with the quality by design for ANDA submission for generics, example for immediate release dosage form. And this is about 164 pages at the FDA site. There is another expectation from the guideline issued by FDA, US FDA, is question-based review. So when we submit any document to the US FDA agencies, they always have various expectations. Some of them I explained to you earlier. Now this is, they say that when we are giving this checklist to you in the public domain, follow this checklist, include that as a part of CTD, Common Technical Document, in submission, so that we don't have to ask any questions to you. And this is what they say in that document, that general framework of science and risk-based approach, and the questions related to the assessment of the criticalities and the process variables, the specifications, and the risk associated with the design of the product, information on the drug substance and the excipients, drug product, the attributes, how you designed this attribute, how did you come to the established condition of these attributes, are there any alternate formulation you worked on? If it is, submit those. How the excipients and the grades you selected for development of the product, and how did you optimize the final formulation and the scale up of the final formulation. In process development, ability to scale up. This is what I was referring. If we are having experiments of 10 kilos in R&D, when you 10 kilos to 100 kilos, 100 kilos to 1000 kilos, how do you achieve? What are your data on that, including stability profile? Impact of raw material attributes, how much this knowledge you got is useful in controlling the risk during manufacturing, during the development of the product, to understand your process specifically at the scale-up level. And as a part of the manual, give the details of the QTPP, CQA, CMAs, and control strategy. CMA is critical material attributes, CQA is critical quality attributes, and quality target product profile and control strategy as a document. Understanding the process, justification of the control strategy, which is the key. And this, when you justify this, they need analytical document. How did you validate it? How did you develop analytical procedure? And nowadays, they expect analytical development also based on quality by design. So traditional versus quality by revision document, they would like to see how do you do assessment of the specification and the quality by design parameters, the CQAs and CPPs, as a part of revision assessment. So if you submit this, they don't have to ask you any more queries. In analytical methods development, the similar terminologies are used as in the finished process form, that with the help of knowledge management, method design, method evaluation for the risk, and the controlling the risk throughout the life cycle of the product, and is the key to support the formulation development for analytical procedure. So in analytical procedure perspective, similar concept, that use it for product development, process development and life cycle. Same concept is used in analytical development as well. So from target measurement, the selecting the technique of measurement, the risk assessment technology, the method development and the validation of the method. Once it is validated, how you can use it control strategy to reduce the risk of the various variables of the methods, right from the analyst perspective, equipment perspective, the ambient room temperature perspective, reagent perspectives, till the validation of getting consistency in the results and continuously improve the analytical method. So you are looking from the perspective of various variables in analytical method, that is instruments, sample preparation, method parameters, and calibration at each of these areas having the impact on analytical data generated. There are certain myths in quality by design which people have, and those also needs to be understood well by the our audience, that it is a quality by design is a systematic approach for the development using scientific and risk management and quality control strategy, which is right. So sufficient details of the development manufacturing information must be included in regulatory submissions. Regulatory decisions must be based on scientific and quality risk management principle, which is right. Another is, which is observed by FDA on regular basis in various countries, is that there is some impression that design space and design of experiment is same. So remember, they are totally different from each other. Design space and design of experiments are different from each other. They are not same. As far as regulatory agency is concerned, design space is not mandatory. But if you establish the design space, it will convince them, it will simplify their understanding that organization has understood the product and the process. That is the advantage of design space. So my personal advice to the various organizations where I am working as a consulting activity, that please implement design space. Don't ignore it because FDA says it is not mandatory. And many people have thinking that QBD is expensive. No, not at all. There are always initial capex expenses, but return on investment is within 2 to 3 years. And then it is afterwards is bonus by way of reducing your rejections, reducing your reprocessing, and in giving you very high productivity and saving the time of equipments, saving the time of changeovers, lot of wastage is reduced. There is also a myth that QBD doesn't change or reduce the regulatory requirement. It reduces definitely the regulatory requirements because you are having flexible operations. You are working within the design space. You have opportunity to move from one normal range to another normal range within the design space. Remember, within the design space. And that is the most important thing. And analytical testing is also important. End product testing is a component because bioavailability, bioequivalence, the BABE studies need to be demonstrated by dissolution testing. Now, these are the things, certain things are required in normal cases, but keep in mind, biggest advantage is your regulatory oversight is reduced automatically when you have a flexible design space document generated in a particular fashion. So overall, when we see the quality by design whole aspect as a process understanding, with the help of desired product performance, process design, performance, product design, identifying the right quality attributes as a critical quality attributes, understanding the right critical process parameters and monitoring them through various control strategies to improve our knowledge, product knowledge and process knowledge for a continuous improvement is the key for the moving forward whole our activity. And then you see here what happens is when we have a product development, look at this definition. Based on whole aspect, the regulatory agencies worldwide accepted this definition of quality by design. What it says is the systematic approach of the development that begins with predefined objectives, emphasizes the product and process control understanding, process control or process monitoring based on sound science and quality risk. Each of these words is highly loaded, you see, and here each of these words is highly loaded. Now, if you see this, what we discussed earlier is a systematic approach to the development. What is that systematic approach for the development is by using different ICH guidelines. First is the ICH Q8R2, that is for formulation development guideline. ICH Q9 guideline, which is for quality risk management. ICH Q10 guideline, which is for product quality management system. And when we talk of drug substance API, ICH-Q11 guideline, and then for life cycle management, ICH-Q12 guideline. All together is helping us to follow the systematic approach for the development of the product, which begins with predefined objectives. And what is predefined objectives here is the expectations of the customers or the patient, what they would like to see the product, how they would perceive the use of the product. And normally when we, as a patient, What do we expect? If I am having, say, pain in the stomach, what do I expect from that? The product that immediately it should reduce my pain or stop my pain. This natural expectation. So that is the objective of the customers, which I have to keep in mind. I have to reduce their pain or minimum pain or take care of them completely. So this is a predefined objective, expectation of the customers or the patients, which emphasizes the product and process understanding. Throughout the quality by design, different steps we have discussed, different guidelines we have discussed by establishing the conditions for design space and with the help of design of experiments. And once we establish this normal operating range, we monitor it on daily commercial manufacturing, which is called critical process monitoring activity, with the help of sound science and quality risk behind, which is helping us for process validation of first 3 commercial batches, and which is in 3 stages. So this I'm going to discuss in detail in the evening. So with this, my focus discussion on quality by design, expectations, and the guidelines, the regulation perspective for quality by design is, I'm done with this discussion on the presentation. So I am now open for any discussion, questions, or if anyone has to share any information, you are most welcome.
Sir, first question is from my side.
Yes, please.
So these days, in academics, since last 2-3 years, we have been seeing that in many of the analytical journals, the application and implementation of QBD and risk assessment and everything, that has been extensively utilized. And they do consider those papers as novel paper, and our conventional OFT approach, one factor at a time, that they do consider, okay, that's taken to be granted, like that. Yes. We received many times the comment that your work has been appreciated, but we miss QBD in your work. And so is there, considering HPLC, HPDLC, LCMS, anything, those are very closed systems, and yes, the chances of variation are very less. Yes. So this QBD implementation in analytical techniques with industry perspective and regulatory perspective, is it that so very much important, or it is only academic perspective that they are looking at?
No, as far as the regulatory agency is concerned, they want to ensure that the parameters which you have, let's say in analytical development, you decide in HPLC method that you are going to use reverse phase. Okay, in reverse phase, the mobile phase which is used there is, let's say, combination of acetonitrile, methanol, and a buffer, say phosphate buffer in combination. And you decided that I'm going to use, say, 30, 20, and 50 proportion. Acetonitrile 30, 20 methanol, and 50 phosphate buffer, aqueous. Now, what is the advantage? How did you come to that range? Now, if you use particular grade of these reagents, is grade making any difference? Do you have any data on that? When you establish this mobile phase, does pH is playing any role? If you say that my pH while using phosphate buffer is between, say, 5.5 to, say, 6.2, for example, and it is giving me sharp peaks during the development of the analytical method of the active substance, separating it from the impurity profile completely. All the impurities are separated properly and integrated properly. So, which pH you will recommend every time as a SOP to be adjusted? Now, this pH of 5.5 to 6.2. So, what is your data at 5.5? What is your data at 6.2? And once you say that I need at 5.8 to 6 pH on daily basis, and if I adjust that, my separation will never get impacted any time for any batch of the same product. So what you are establishing by using design of experiments there, that my range of 5.8 to 6 is daily implementation, that is a normal range. However, if I find that after 6 months, I can change it for reducing the retention time and improving my productivity. In one day, instead of testing 10 batches, I'm able to test 15 batches. By changing the pH, I'm reducing the runtime, and still I'm able to separate all the impurities, and the sharpness of each of these impurities and the active is maintained. I am changing to the new range now, which is allowed within the design space. You don't have to take permission to change this of regulatory agency at all. That is the biggest advantage, which normally you are not able to achieve. This is one small example. So this is how we find out various variables in method development, right from analyst, environment condition, room conditions. Say for example, acetonitrile and methanol, after a few hours while using, it changes the pattern because evaporation has an impact on the room conditions. How you are going to monitor it? I had to ensure my temperature whole day should be between 22 and 25 in the room. If it goes beyond, I am having a problem. So you have to ensure in your SOP that my temperature should not go above this in any case, which will have an impact on my analytical risk, my critical analytical parameters. Critical analytical process parameters are affected. So the variable we have to identify with the help of fishbone, and those have to be addressed with the help of the DOE. Establish it, then you forget whole life on that. So that's the biggest advantage you get. You are correctly identifying all the possible variables through the risk assessment, which normally we don't do. Normally, as a knowledge expert, in our head, we have everything back of mind and we play with that. But now here we have to put it on paper with the support of the experiments, establish it. That's the whole advantage of following quality by design. Otherwise, normally what was happening till today, if you see even United States pharmacopoeia, there are various validation parameters. 6 different elements are there: linearity, limit of detection, etc., etc. Now, USP is also talking on the analytical method by quality by design, the latest document, as a part of general notices. So it is the key thing for the submissions. But more on that, Whenever I get an opportunity, maybe after 6 months down the line, I can explain to your students in person because it's a critical for them to understand this. We in the industry, when we get the fresh graduates, we have certain expectations, doctors. And when we ask certain questions, they are not able to come up to the expected level. And there are big gaps there. So this, we will have to spend time with them and with the real one-to-one and explain to them what do I expect in the industry from the fresh graduates. If I have to discuss only this, your question, at least I have to speak minimum half an hour.
Okay.
So that they can grab it and answer to the interviewer for at least 5 minutes.
I purposefully asked this question. The thing was, that was with reference to our previous submissions, but yesterday only, or day before yesterday, we had a pharmaceutical quality assurance presentations, student dissertation projects. The examiner and almost all the students worked upon QBD, risk assessment and development of method through QBD and MO, the design space validation and everything.
Right.
The examiner was very curious. Examiner asked the same question to all the students. Really, it needed, what is your purview? What is your, it was a very common question. Few students worked extensively and they could be able to answer. There were few students, they were fumbled in between. But now awareness has increased in field of analytics also. Like we are receiving the comments from the referees, but still, sir, still there is a session in analytical chemistry. I'm not a person from analytical chemistry, but I do work. There is a session in analytical chemistry. There are few people in analytical chemistry. They still feel that resolving a compound from a simpler metrics like tablet or pharmaceutical ingredient, where you are not working on impurity profiling, just simply percentage assay determination or anything, where implementation of QBD is just like, you are sending a whole battalion for killing an ant. It is one factor at a time, and you develop the method and validate, that is sufficient. And exactly the thing which my students in their seminar were depending. But anyways, what you said, that is application of design space in modifying the method without regulatory hurdles. That is something that is actually the key component where it can be defended. Truly, sir.
It is essential, doctor, to identify these variables. And here normally we don't do that. And so when we, even today, I always call, when you do one factor at a time, experiments, one factor at a time doesn't give you optimized data. When you have 2 or 3 factor changes in one experiment, it also studies the incompatibility and of various variables. And that gives you a maximized data. That is the key. So that is where we say that factorial design. 2 factors or 3 factors at a time or 4 factors at a time. And I generally advise the organization, don't cross your limits of 3 factors at a time, because unnecessarily you are increasing the complications and number of experiments, which could have been either, if you are having 2 factors, then only 4 experiments, high and low, 3 factors, 8 experiments. Try to minimize the experiment, but achieve maximum in minimum experiments. In normally, what we are doing today without quality by design is 1 factor at a time, trial and error, nothing beyond that.
Sir, there are some questions here. Can you be able to see the Q&A, sir? There you have questions itself.
Let me open it.
Is it?
Yes.
Professor Rashmin, he is faculty here.
Yes.
He has 4 questions.
I would like to ask that, as compared to the traditional approach, QBD is speeding up or slowing down. This is again a Dr. Rashmi. This is a myth again. Okay. It doesn't slow down. Remember, since you are studying various factors at a time in experiments, it reduces your number of experiments in comparison to trial and error, one factor at a time. That's a fact. It reduces your experiments. You are able to launch your product much faster than the traditional way. Your second question is, are there any specific regulatory requirement concerning documentation for risk assessment? Yes, it is there. It is a part of QRM. It is a part of the Q8, Q9 together. You read it. It is there very much. And please refer. I have given you the names of the documents, development of the product, how to do by US FDA. There are 2 documents available on the website. One for immediate release, another is for modified release. Please refer it. You will get this complete information there. Same applies to your real-time testing. Okay. Now, real-time testing, there's a part of your question 3 is process analytical technology is a key. And this I'm going to discuss in the evening. To some few minutes, I'm going to spend on that it is based on process analytical technology. And the 4th is the regulatory challenges. This also I'm going to, some of these I discuss with you, what we expect for, what FDA expects in QBD approach implementation. Some I'm going to discuss more in the evening. So I hope you will be there, Professor, in the evening. The next is Baya Subbarao. No problem, you joined late. Okay. I am faculty in so-and-so in Hyderabad. Okay. Give some information on lux hours relating to the photo stability. Okay. This subject of Q1 and Q4 is not a part of our subject today. We are focusing mainly on quality by design and not on the ICH stability guidelines. So that will have some other session separately. There is an anonymous attendee which says, how FDA is considering artificial intelligence? Yes. This is a good question. Okay. Now, artificial intelligence work is on. To some extent, the work is there. I'm going to touch upon this in the evening. But artificial intelligence is going to guide us on planning of the experiments, which parameters we should address, and they will guide us. But however, no artificial intelligence can beat human intelligence. It's also true. So we have to keep in mind, whatever data we feed to the computers or the robots, they only follow that, nothing beyond that. Whereas human brain is so far, human brain is much better working than the robots. I have seen recently a video where artificial intelligence is used for the training purpose, which is very interesting, which I liked. And if I am permitted, I will be very happy to show you that video in the evening.
Sure, sir.
I will be very happy, and it is excellent where the world is going and where are we. So I am going to discuss that in the evening.
Sir, one of our faculty member has taken up one project on predicting compatibility of drug and excipient and developing data for artificial intelligence.
Yes.
One research scholar is he has started working upon.
Yes.
So probably I'll call him. He'll interact with you also, because this is rather it is seen as a future.
Sure. Sure, sir. It will be a pleasure to interact such talented students. Even I am learning a lot, even today, at the age of 73, from youngsters. They are future of our country, future of our industry. pharma industry. Today, pharma in India, as rightly mentioned by the Prime Minister, is the pharmacy of the world. So these, our youngsters coming out from various universities, from your university, your institute, are future of our pharmaceutical industry. So I also learn from these youngsters very much. Every day I learn.
Sir, the another question is from Dr. Rashmin that you have, I think so. There are some questions which are in chat also.
Okay, let's see.
What to do if even after applying the QBD approach in analytical development, SST is failing at the verification or application stage?
Oh, okay. Yeah, I mean, see, these kind of questions will always come up even otherwise also. So what we do in normal cases, forget about QBD. Your trial and error method, develop procedures without QBD also likely to fail. So what we do? So we have to rationalize our thinking. Important is how rationalized we have, what kind of strategy we have used to identify the variables. Have we done correct job, right job in identifying proper variables impacting my process? So that is the key to understand whole risk management. And the development of quality by design revolves around the nucleus of risk management. ICHQ9 is the key. How do we identify the risk and why? Now, many times, Doctor, I ask this question to my audience in various my commercial assignments. Is that because this is a straight question and everybody can answer also, is when you come out of your home, how do you cross the road? And why that way? Who taught you that? How to cross the road? As simple question as that. And what happens if you fail in that? so first thing is, the first guru in our life is our mother. Remember, there cannot be any better teacher than mother in our life. The first guru. And she holds our finger when we are small, 1 year, 2 years, 3 years, and walks on the road, always guides us how to walk, what to look for, what are the risks involved if we don't follow the system there. And slowly we start implementing that, keeping in mind mother's advice. Because we realize, yes, the car is coming from one direction with a high speed. So I have to take precaution and I should use zebra crossing. Zebra crossing when there is a green light only for walking, not for green light for the vehicles. So it is important for me to keep in mind that I have to follow these steps and for my safety. So what are the variables here? Is the road is wide, traffic is there, and also there are risk takers. Some people want to run to cross the other side. They enjoy that as a fun. And in that process, anything wrong can happen to that individual. All these risks we have to keep in mind. So when to cross? If there is a overbridge, most safe way to go. So all these things we keep in mind from our childhood. Nobody teach us this. And knowingly, we take the risk because the vehicle is far away and I can easily cross. So that's a risk-taking decision. Sometimes you take it in product development, sometimes you take it in analytical development. Some risks are built in, which we always call as residual risk also in the development, which we successfully monitor. Though these are critical quality attributes, we say, yes, I am buying the API drug. Every time the particle size is critical, I am checking the particle size. It is critical for dissolution. It is critical for content uniformity. But I am not, till the time I do not check every corner, I am not assured every container is same particle size. So the risk remains. When I say the identification is a risk, I am saying yes, it is a risk, but I am taking the sampling of statistical sampling, identifying and minimizing the risk. So that remains a CQA, but I don't have to worry about it because my statistical sampling is helping me. Over and above, as per the law, every container of active drug substance I have to identify before use. So I'm taking these additional precautions. So that risk goes down. So I don't have to worry much about identification perspective. That's how we look at it. Whereas particle size doesn't help me from that perspective. I have to ensure every container at different stages, different layers in one container, same particle size is there. So that's a different aspect. Impurity profile is a different aspect. Process related impurities, product related impurities have a different approach. So what happens if it fails? There is no answer from that perspective. Whatever happens in traditional way will also happen here then, because we are ignoring certain areas. We did not develop the product in a right perspective. Okay, then what else is how to decide on what percent should be used in any formulation, any dosage, also in QBD. This question looks to be incomplete rather. I don't know what the Karnav Dave wants to ask. I don't know. I couldn't get it here. Maybe he is saying that what percentage of excipients or what? I cannot understand this question. Your audio is not there.
Sir, there is a question from Dr. Fatima from KLE University. She's specifically asking about the appointment of a statistician to interpret QBD data. Yes, it's a big yes, I think so.
You know what happens? I'll tell you. Some people want to go for cost saving. Some softwares which are available for design of experiments and design space understanding. Some are only DOE software. Some are beyond the capabilities of DOE, which we need today for a statistical data interpretation. Now, this data interpretation, it is always preferable to have a statistician at site, at least one person, and who can help to explain the outcome of the data. So it is most welcome recruitment to have such a statistician who understands the whole pharmaceutical development and the quality by design concept. But this, for example, you have a Minitab software, it has extra capabilities, Minitab, and these statisticians has a tremendous value there to support the development of the product and the method as well. So they are a great use to us.
So there is another question from one of the students, that is fundamental question related to quality risk management, that how the risk can be assigned number 1, 2, and 3 is very fundamental, but we are eager to listen from your side.
Risk can be assignment.
Risk 1, 2, 3 scale be assigned.
Oh.
That is very fundamental, but we would like to listen from you.
No. See, there are 1 to 3 scale if we are giving. What happens is we have to keep the relations in mind of the variable on the quality attributes or the process parameter, critical process parameter. Now, there are 3 types in this. One is the impact of the risk. Another is probability of the risk. And third is detectability of the risk, which we call it as a risk priority number, multiplication of the 3. And this risk priority number for each of the CQAs or the CPPs has to be identified. And your initial risk assessment, and based on the control strategy, how you minimize the risk. So if you come from the number 3 at each of these, impact, severity, the probability and the detectability, 3 of these, it is 9. 9 into 3, 27 is your RPN maximum risk. And if you are, by having a proper control strategies on the key variables, you are saying now my amended risk is 9. I have reduced the severity. I have reduced the probability of happening it, and more the focus should be on the probability. Severity may remain the same, but probability can get reduced to a large extent by your control strategies. So your number may, if suppose it has become 9, so you are saying now my amended risk is 9. That shows that your control strategy is proper and it has gone from red 27 number to now yellow or green color. And that you have to maintain that status throughout. So severity, when we say 1, 2, 3, 1 number is low. Second, medium number 2 is green, that is, yellow, that is medium risk. And third, red is high risk. So low, medium, and high. This is how these risks are identified, low, medium, and high.
Sir, there is another question from, again, it is from student side. Why QBD is not incorporated in pharmacopoeias?
These are the guidelines. Pharmacopoeia is mainly for the monographs and the testing procedures. And this QBD is for the development of the product. So pharmacopoeia is for the quality of the product, which are commercialized. Whereas quality by design is for the research and development department specifically to develop, which is to be implemented on manufacturing scale by manufacturing team, by quality assurance team together, and which is measured by quality control team.
There is another question, and probably this you are going to include in the next lecture. That is role of QBD in regulatory submission to the agencies. I think you will discuss in the next session about this.
Yes, in the evening.
We are not going to take this. Another one is, what are QTPP safety and efficacy required? Sir, this question, there are 3 questions here. Bachani Priya, I think so, and the question, it is in question answer session. And probably, sir, this with your consent, we can take this as a last question.
QTPP safety efficacy required. This is I'm talking in the evening. I'm focusing this in the evening. And if I forget, please ask them to remind me again.
Okay, sir, then I think so most of the questions are answered.
Okay.
With your permission, shall we conclude this session?
Absolutely fine. You will have to decide on that.
Thank you very much.
Because it's so long time, like 2 hours or more than that.
No problem.
Please, Jagruti madam.
Yes. Thank you very much, sir. And thank you all the participants. So with this, we end up with the first session, and we will continue with the second session at 3 p.m. So all the participants are required to join before 3 p.m. Thank you.
Okay,
Thank
bye.
you, sir. Nice time, sir.
Bye. Thanks a lot. Bye.
Thank you, sir. Hello, sir. Good afternoon.
Good afternoon. Good afternoon. How are you?
Yeah, fine, sir. So relax, sir.
So,
Yeah.
over the morning session.
Yes, sir.
Are you able to listen?
Yes, sir. I'm audible, sir.
Are you able to listen?
Yes, sir. I'm audible.
Hello.
Yes,
Hello.
sir. Yes, sir. You are audible, sir.
Hello. Are you able to?
So we can listen, but I think so.
Good.
Hello.
Yes.
Okay, sir. Fine.
The video, sir, if you can delete afterwards from the presentation, I would appreciate, because those are copyrighted videos.
Sure, sir.
You need to delete the videos afterwards.
Sure, sir. Will do.
You are in Anand, is it?
Yes, sir. We are nearby Anand, in between Anand and Nadiyad.
There is one more, Anand Pharmacy College.
Yes, sir. Anand Pharmacy College, that is situated in Anand.
They have invited me for a talk on 19th.
Okay. See, actually, sir, what happened in last 3-4 years, MPharm in regulatory affairs has been picking up the popularity.
Okay.
We had this program, Pharmaceutics Regulatory Affairs, since I think so 2011.
Okay.
But since last 3-4 years, students are preferably coming for. Initially, the time was when the admissions were not available in QA or technology. Then the students opted to go for this regulatory affairs. But now students, they do come preferentially for RA.
Okay.
And there are many institutions now, they have come up with MPharm RA.
They want to become inspectors.
Want to become inspector, and personally, I feel the experimentation is not there in RA. That is even one attraction. Because in QA, Sutics, heavy experimentations are there, but in RA, that is missing. Paperwork is there, rather, no, salary structures are almost similar, opportunities are comparable.
Yeah.
So, sir, shall we start?
Yes.
Okay. Please, madam.
So, welcome once again to the Charuset Endowment Chair on Regulatory Considerations for Pharmaceutical Development, a Global Scenario, organized by Ramanbhai Patel College of Pharmacy and T.B. Patel Center for PG Studies in Pharmacy, Charotar University of Science and Technology. For second session also, we have with us Ranjit Barshikar sir, and he is the CEO, QBD International United Nations and Advisor, the Geneva Editorial Board Member, Journal of Generic Medicines, UK. He has an experience of more than 51 years, and in the pharma industry, especially in the quality management, R&D, manufacturing, US FDA, EU, WHO, etc. compliances. He is also an expert on quality by design, data integrity, CAPA, quality culture, and various other CGMP subjects. So I request Ranjit Barshikar sir for the second session on QBD compliance, the current scenario and future challenges. Sir, please.
Okay, thank you very much, madam, for the kind introduction which you have done. Friends, we discussed in the morning about various guidelines and the regulations on quality by design and some part of what regulatory agencies are expecting us while implementing the quality by design perspective. So now we are going to talk on the current strategies for the development of the product and now the changes while doing QBD implementation, and after that, what are the future challenges in product development and what kind of role automation will play in the pharmaceutical development and the organizations in general as such. I'm just sharing my presentation now. What is message I am getting, madam, is you cannot start screen share while other participant is sharing. Take care on that.
Yes, sir. Yes, sir.
And let me know once it is through.
Yes, sir.
So now you can share your screen.
Okay. Good. Thank you. Good. Are you able to see full screen?
Yes, sir. It's fine. Perfectly in full screen and visible.
Great, great, great. So the friends, in the morning we spent some time on learning on the regulatory perspectives of quality by design, and let's go a little deeper in that and what kind of current scenario and what will be future challenges in the development of the product. And we'll spend some time on the automation perspective as well and various technologies which are going to give big challenges to us in the pharmaceutical development and manufacturing and the testing as well. So let's understand first. Now, before we go to the actual subject, a little bit background, a preamble is required for the audience to understand what is the difference between generic drugs and branded drugs and or a patented drug, and why it is generic, why it is branded. So just to quickly take you to the branded drug is a patented drug or a organization which is introducing that drug or a finished dosage form into the world market, and they have around 20 years of patent life. Around that, in some cases it goes 2 years beyond, in some cases it is slightly less as well, but by and large it is like a exclusivity to market in the world, and that is why those are called inventor's drug or a patented drug or a branded drug. Now this patented drug is for the whole process which organization files with the various countries in the world as a patent, and which you cannot copy any of their processes. If you copy it, you can go through a legal procedure for infringement of the patent. That's the law in America and several other countries where patent regime is being implemented very seriously. Now, from quality perspective, generic drugs. Now, generic drugs are almost same or identical with patented drug, and this development of the generic drug, you are not going into the basic research. You are picking up the information available of the patent drug and trying to copy the product, but some variation during manufacturing so that you do not come under infringement. Now, when you follow this, there are so many critical aspects to this area, legal aspects to this area, and every organization has intellectual property, intellectual proprietary department, IP department. And these are the legal departments who knows the details of various products which are filed in various countries as a patent. And this patent is for what? Which area the patent is holding in various countries. It could be different in different countries. Now, generic drugs are focusing to US market. So, for example, 99% of Indian companies who are exporting the product to America are generic companies. They are copying the patent product after the expiry of the patent, and they can launch the product in America as a generic drug, same product as a generic. Now, when this product is going to be as generic, there is an advantage to American citizens. What's that advantage? That it's a cost benefit. Now, North America, which includes Canada and Europe and few other countries, they have a system of very strong insurance support to the patients. And this insurance takes care of all the payments of the patients in the hospitals. And when that happens, insurance companies, if they are paying the insurance of a patient who was using rather a branded or a patented product, the cost could be thousands of dollars to the insurance company. But when the patient uses the generic drug, the cost to the insurance agency goes down to around 50 to 60% of what they could have paid to patented drug user. Now, but what is the similarity then between generic drug and branded drug? And why generic drug we have to develop in a particular way for American market? So what is the whole concept of quality by design, what we discussed in the morning? That supports the generic drug development to ensure that the generic product is identical to the branded drug. Identical in what? In dosage form, in safety, in efficacy, in route of administration, identical in quality as well, and performance characteristics and the intended use by the patient. So, quality-wise, safety-wise, efficacy-wise, there is no difference between generic product and patented product. And you are legally and officially allowed to develop generic drug copying the patent product. Now you can copy as per the open portion of the patent. Secret portion is certain key steps which they don't disclose in public domain. the companies. That is their trade secret, and they won't like to disclose it. Whereas the clever scientists try to do the best to develop a product equivalent to patented product or a branded product. So once we understand there is the generic drug is the same as patented drug, things are simpler. The benefit is to generic companies as well, because cost of development is very low than patented companies' cost of product development. Multinational organizations by and large spend millions of dollars to develop a product over 10 to 15 years time, whereas generic drug developer need maximum 3-4 years to develop a product and establish that it is bioequivalent to the branded drug. So if you see in nutshell, generic drug is of same quality and performance as branded or a patented product. In America, 80% of the prescriptions today, 80% prescriptions in America are of generic drugs. And that is a big advantage in America. That is an advantage to generic companies as well. They get very good business in America. And that is why every Indian company who are interested in export, they go for US FDA approval, they go for US FDA inspections, and they go for US FDA marketing their product as generic drugs. So with this background in mind, then coming back to our subject is easy. Now, what we were talking in the morning on quality by design, which is required for ANDA submission to US FDA. ANDA is abbreviated new drug application for US market submission with US FDA. Now, what is happening traditionally? There are number of variables in manufacturing of the process. Variable of the input material, the suppliers, as I discussed in the morning about particle size, variation in particle size from one supplier, container to container. So there are several such issues. That was one example I gave you. Some suppliers give slight variation in appearance also. So we have to keep in mind the variable of input will give output also as variable output. In other terms, we say that garbage in, garbage out. Whatever you feed the quality, you will get the same quality when there is output. In other words, so variable input, there will be variable output because the processes are fixed. And the whole document of manufacturing is standardized, that you mix 100 kilos in a mixer grinder for 10 minutes at a per RPM of 15 RPM. That's the standard written there. The operator has to implement exactly that, nothing beyond that. And this is how unchecked processes, the fixed processes with the fixed variables inside was going on, and that was not giving us consistent product quality. So locked process variables always causes variability of the raw material to become variability of the drug product as an output. Remember, garbage in, garbage out. So we have to keep in mind, traditionally we are having a fixed process, having number of variations in the various attributes. So then why a quality by design? So current we have seen, we are having variability is an issue. We saw that variable input gives variable output. So we have to find out how to control these variables, how to minimize these variables during different stages of manufacturing. And that is a skill to be achieved by quality by design. And this variable, research and development department has to look into the perspective of keeping flexibility in the manufacturing process to handle such variables in a effective manner. So variability or the variable is the key thing, and quality by design is helping us to minimize the problem of variables, minimize the variability in manufacturing process, right from input to the output. So then why FDA went into quality by design? As we are doing today, FDA was doing this for years and years, and so many decades as well in the past. There were several issues of product quality with the patients not getting proper relief after using some products from some organizations. So FDA, US FDA tried to analyze why there is a shortage of the product in the market. After doing the survey, they realized this shortage is happening due to several recalls of the products from the market because of quality issues. Then they started discussing with those organizations who were recalling the product, and based on that, they realized these quality issues are happening because of several variables, and these variables needs to be addressed, and the industry needs to be guided how to address these variables issues during development, during manufacturing, during testing as well, keeping in mind patients. And that is why they thought, let us introduce quality by design. Another was, they got an impression that the processes which are developed by the industry are non-robust. They are not based on strongly on the science and risk base, and hence has a frequent failures, frequent reworking, reprocessing, or rejections. Why that can happen? Because some organizations are in a hurry to file the document to become first to file the product in American market. So they follow the shortcuts, cost cutting due to shortcuts. They don't spend sufficient time in product development. Some organizations have lower investments, so they cut corners, they cut the costs of development, which results into the non-robust processes as well. So this is also one of the reasons of the non-robust processes, creating more variables in manufacturing and more rejections of the batches from the market, resulting into the shortages in the market. So how the current scenario is, vis-a-vis quality by design. So what was happening is, till today, it was traditionally quality by testing, QBT, T for teacher, quality by testing. And now we are considering advantages of quality by design over quality by testing, QBT over QBD over QBT. So let's see what are those. Some differences in both. So traditionally, product development is trial and error. Quality by design says we are systematic process approach, as we discussed in the morning on guidelines with the ICH and the FDA document. Multivariate experiments for design space, design of experiments, and knowledge driven. Manufacturing process traditionally is fixed. By quality by design, it is flexible, and within the design space, you can have a lot of flexibility. Process control fixed. In QBD is real-time monitoring process of all critical parameters. Specifications in traditional way is limited to the batch information only, whereas QBD is overall quality performance and controls based on the product performance, process performance.
Sorry, I missed that. Could you say it again, please?
Yes.
I thought so.
Okay. So I'm coming back to the specifications of traditionally. We have a fixed specification where we have limited information about the batch, only very few information of few in-process checks. And at the end of the product, it is a testing on the specifications, nothing beyond that. I will give one example, is that if my product is injectable and my batch size is 1 million vials injection, batch size, one batch, 1 million vials. Traditionally, I am testing the sterility as per the pharmacopoeia procedure, taking 20 vials out of 1 million vials and doing the sterility. And when I do the sterility, based on 20 samples data, I am saying batch of 1 million vial is sterile. Today, we are building the process in such a way that I am ensuring now every vial is sterile. There is no doubt. I am confident on that. That I am developing quality in designing the product, not only testing. That's the difference. Improvement of the process is continuous and throughout the life cycle. And of course, regulatory oversight for traditional way is inspection and reviews, and for any manufacturing changes you are doing throughout the life cycle. Whereas here, there is a flexible regulatory oversight in QBD based on the risk assessment which is being submitted, and every year data which is being given to the regulatory agencies. So now no more quality by design testing. It is about monitoring all critical steps during manufacturing, which is quality by design, designing quality into the manufacturing process. So it involves, QBD involves focus on patients, minimizing or eliminating the variables, key variables, monitoring the variables and zeroing on the variables, eliminating the variables. We are focusing on the product uniformity. Because of the process, if it is variable with the PAT, we are process analytical technology, which I'm going to discuss subsequently more, is I'm bringing uniformity in the output or consistency in the manufacturing. Though some variables are as in input, but I'm monitoring in such a way that variables are reduced or eliminated. Everything is science and risk-based approach, and which helps me for continuous improvement. Why it helps me for continuous improvement? There was one question in the morning, one of the participants asked, feedback and feed forward concept. So here it is, you see on the screen, is whenever while implementing and this quality by design in manufacturing, we have already identified critical quality attributes and critical process parameters. And these monitored successfully with the right control strategy is giving me a product which is having consistency in measurement, consistency in quality, and which is validated, which is called process validation. And there are 3 steps in process validation, which stage 1 is design, stage 2 is qualification, and stage 3 is CPV, continued process verification. Now this CPV data, which is generated, is given feedback to R&D by manufacturing and quality assurance team, and R&D collects the data of 25 batches or 50 batches or 100 batches, and they look at all the critical parameters in that data and see whether they need further improvement in the process. If not, they continue. But there is a record saying that the data was reviewed after 50 batches, and if there are any changes required for, say, example, yield of the product, by changing some parameter within the design space, that changes will be made, and there is no need of regulatory permission for that because you are within the design space. And this whole continuous improvement is throughout the product life cycle. And that is why we discussed this in the morning, the definition of ICH guideline for quality by design is a systematic approach to development of the product which begins with predefined objectives, emphasizes the product and process understanding and process control based on sound science and the quality risk. So always remember this, each of these words are highly loaded. Product and process understanding, process monitoring, sound science and quality risk management activity. Now, these terminologies in quality by design, remember this whole life. Remember, though you go to any section of pharmaceutical plant or biopharmaceutical, remember this terminology all the time. Short forms and the long forms. QTPP is quality target product profile. CQA, critical quality attributes. CPP, critical process parameters. CMA, critical material attribute. DS is design space. CS is control strategy. So whole activity of quality by design during the development is of these 6 steps. And the next is life cycle management. So keep in mind these 6 elements of quality by design: QTPP, CQA, CPP, CMA, DS, and CS. So how we should proceed for that? When we would like to go for a roadmap, how should we act on that? So for that purpose, when we move into the implementation process for quality by design, remember, we are starting with the target product profile, QTPP. From QTPP, we are identifying the critical quality attributes based on the risk assessment. Similarly, from material attributes, we identify CMAs. From PPs, that is process parameters, to CPPs, and all those CPPs identify design space, control strategy, and then life cycle management. So this is a whole roadmap for implementing quality by design concept in product development and implementing in manufacturing and testing as well. So what are we doing there and why? So here we are linking patient, product, and process together. Remember this. What we talked of is quality target product profile. What is needed to the patient, right? We discussed this. Target product profile, quality target product profile is based on the patient's need. And we talked of this, I will show you in one of the slides, but why it is important. From risk management handling perspective, at every stage, we are seeing the product, process, and facility, or facility moving towards process to product to patient. And that continuous feedback requirement back to facility and to the R&D for further improvement. So process parameters, various models to be used in process parameter identification and design space monitoring, listing the quality attributes, justification of quality attributes, justification of process parameters. Everywhere we need to identify the risk where it can fail and successfully implement risk management strategy to mitigate the risk. And that is how it is linking the process and product to the patient. So what patient needs then? What patient needs? Simple. What patient needs is safe and efficacious product. As simple as that. That they want value for money. If you are saying my tablet or capsule or a dose contains 100 milligram per unit or per tablet or per capsule, and they are paying you 100 milligram per capsule or tablet. So you have to give them back 100 milligram during the shelf life, not on first day. Till the last day of the shelf life of a product, they must get it. There is a range for, say, for example, assay or the potency, depends on the in domestic specification or pharmacopoeial specification or regulatory approved specification. If suppose the approval is for 95% to 105% of the potency or active drug per tablet, it means minimum 95 milligram per tablet of 100 milligram on the last day of the shelf life. Remember this. So they need safe, efficacious product, no side effect or lack of side effect, and the dosage acceptability, route of administration, and the frequency of dosing should be informed to them from the organization perspective. And this information is built into the quality target product profile. There we identify the CQAs, and all the CQAs are also dependent of the product, dependent on the API CQAs, as we discussed earlier. So overall, if you see quality target product profile, which is the first step in the development, we define in advance what kind of safety patient needs, what kind of efficacy they want, what kind of quality they want, and also manufacturability. We have to focus develop a product which is your factory can manufacture. It is of no use if the factory doesn't have capability to manufacture and you develop the product for that. So remember, the R&D team has to work in tandem with manufacturing, depending on the type of equipment, depending on the type of environment condition in manufacturing and the facility, they have to develop a product. So safety, of course, efficacy, safety is very important for patients. So how this branded drug is getting identified is also reference listed drug. There is a book available in United States, which every physician, doctor refers also. And frequently you will find it with the doctors, practitioners, pharmacists, as well as the organizations, manufacturing organizations, where this patent drug information is given on physicochemical characterization, product development, dissolution method, clinical data, and PK data. So whole this information is available in that. Is published document, and anybody can buy it and have access to the information of all the patented product or the products which are existing in US market. So they are innovator product. This composition is studied while doing reverse engineering for a generic development, and the same with the prior knowledge with the company has in generic companies, and the labeling information is already available based on the product availability in the market of the patent product. Here, this RLD book or the physician desk reference book, PDR, has information on the excipient also, what excipients it contains, but they don't give quantity of excipients. So generic companies have to work on the quantity of excipients. So based on the background information, the organization has to generate based on the patient need and the branded drug or a patent product drug or a reference listed drug in the market, which you can purchase and study that. So that everything can be built into quality target product profile. Remember, and this is strongly recommended by FDA for the clinical development, drug safety, manufacturing, marketing, regulatory, R&D, and quality before and during development. So it is very, very important to have a right document in place for a QTPP. One example I'm showing you of the QTPP, which is immediate released drug. Look at this example. This will be a good guideline for the development as an example or a case study. What are the QTPP elements? Dosage form, design, what kind of dosage design, route of administration. Say for example, here it says tablet is a target. Dosage form, tablet target. Design is immediate release. Route of administration, oral. What is the strength? 20 milligram. So the element and the target of the element has to be defined in advance. Then there are specifications of the product, the stability of the product, at what shelf life, 24 months at room temperature, the product is stable. Packaging material, container closure system, labeling, and if there is any alternate method available for administration to the patient. And in the third column, we have to give justification of each of these, why you decided this. So that way, the things are fairly simple if you follow this example generated by FDA themselves. So we are transitioning from high variability to low variables, low variability during product development. That helps us to enhance the product and process understanding. And from compliance perspective, we are moving towards science and risk-based product and process understanding, better enhancement of our knowledge of the product and process, and design the product for effective and efficient process, and also real-time quality assurance. What is real-time quality assurance? It means monitoring all the critical parameters on the spot where that process ends, that process steps ends. And it could be part of process analytical technology or some other techniques where you can implement online for knowing the results of the particular step to move to the next step. And this is what I discuss with you, feedback and feed forward process. Now look at here what the patient wants. Let's take the example of the tablet. And a patient is having running nose, severe cold. Even if we have, every one of us have gone through this. When you have a severe cold, running nose, what do we think? Oh my God, it's a big headache, now it's cold. I'm having running nose. What should I take? I must get quick relief. So for me, quick relief is important as a patient. But how do I get quick relief? From a good product. Now, good product is what for me? What is critical to me? Is it appearance critical? Is it hardness critical? Is it dissolution critical? Is it content uniformity critical for me to get quick relief? Remember this. So when I talk of quick relief, for me, as soon as I take the tablet, it goes into my stomach, it disintegrates faster, and particles of the active drug are available for taking care of my running nose. Now, if I give the product which has an effect after 1 hour, that is not the right thing. I must give the product as an organization which can have very good dissolution characteristics. Generally, for immediate release products, the maximum is 30 minutes to 45 minutes dissolution period as per the specifications in the pharmacopoeia. But for the filing documents to American market, you may have a different parameters, which is stricter than the pharmacopoeia, which is stringent than the pharmacopoeia. So dissolution could be, say, maximum 30 minutes. So in 30 minutes, at least up to 90% must be available, so that I'm calling myself to be quick relief. But better than that, in 15 minutes, if I get 60 to 70% dissolution, I'm sensing the relief. My running nose is reduced. So that's where I'm looking for right dissolution. And the content uniformity. So content uniformity means every tablet I must get definite percentage, let's say 90% of the drug. Every tablet I must get it. Minimum 90%. So that's the content in each tablet. So every tablet, there will be minimum 90% or 92.5% or 95%, whatever is given in the specification, given in the filed specification with regulatory agency. So for me as a patient, what is critical? Dissolution, yes. For content uniformity, yes, critical. But appearance, no. But important is dissolution and content uniformity are more critical. Hardness, I can manage online. The dose is not a question. It is there definitely, 100 milligram per tablet. So what is most critical here for me from quality target profile is dissolution and content uniformity. Remember, So what is the relationship now between CQA and CPP? Let's take the process of mixing online. What we look here is a blend uniformity. What is blend uniformity? Is active drug is distributed homogeneously throughout the blend material of 100 kilos in a container. Before I move to the next step, before I compress the tablet. So I have to establish assay. So that is called CQA, critical quality attributes. And what is the process parameter? Is in such case of process step is mixing time, mixing speed are critical for me in the process mixing. So if I handle time and speed properly, establish the design space here properly, I am going to get the right potency. So I am matching the requirement of CQA. And that is where we say the ability to control and monitor the mixing time and speed is a critical aspect of process step of mixing. Same way, let's take the example of depyrogenation in sterile area, in sterile manufacturing injectables. In depyrogenation activity, means those vials which are going through the tunnel of depyrogenation around very high temperature, all the pyrogens, if anything is there, gets completely removed, killed during this process, and the container is completely depyrogenated before filling the product. Same thing with the material. So material also, there are different processes where you can ensure that it is the pyrogen free. There are procedures for that. So how do I know that I am successfully making it depyrogenation? So I have to ensure there is no pyrogen at all in my vial. The presence of pyrogen is not there. So there are test procedures. That is CQA. And what is CPP there? How can I know that I am going to make CPP a critical process parameter working well? Is on how the vials should move on the belt, the speed movement of the vial and the temperature used in the tunnel. If I do it hurriedly to increase the productivity, there it is likely that some vials may fail in pyrogen testing. So we have to monitor the speed and temperature to establish and ensure every vial is pyrogen free, which is critical to the patient, which may have a severe adverse effect on the patient. Therefore, pyrogen is a critical to the patient. And that is where we talk of handling the risk assessment is critical throughout the product development and manufacturing, is right from handling the risk of the knowledge gaining throughout the activity. If you don't have a proper knowledge, there is a big risk. That knowledge or little knowledge can impact the process development. So we have to take care of that risk first itself. I must have a proper knowledge. I must have a team of a right knowledge on the QBD concept and the requirement of the QBD development and the chemistry of the product, chemistry of the drug. So knowledge is the key thing and one of the biggest challenge in industry even today. Then the process knowledge, everywhere we need to handle through the risk, find out what can go wrong in my process and what impact of that wrong will have on the quality of the product. Design space, control strategy, and verify the process. Handle it in the same process, same activity in the analytical area also throughout the life cycle for stability studies as well. Now there are different tools for risk assessment. Is a fishbone or 6M or cause and effect analysis. Second is quality risk assessment. It's a qualitative risk assessment where we say is road signals lights is high, low, medium, and FMEA failure mode effect analysis is quantitative risk assessment. So fishbone, qualitative high, low, medium, and failure mode effect analysis, quantitative risk assessment. There are several more tools also for that. Now, one example here I would like to show it to you is a risk-based approach for initial risk assessment, which I was briefly talking to you in the morning when we are talking of CQA against the raw materials and the process step, different processes. So raw material can affect on identification, on assay, on content uniformity, on impurity, on dissolution. If API supply itself is bad, all these parameters will get affected. All these CQAs of the product will get affected. In blending, content uniformity and dissolution. And in compression, dissolution will get affected because if the hardness increases, dissolution will get delayed, and the delayed dissolution will affect the patient. They won't get the effect of the product as per expectation. So we have to address these kind of things in such a way with the right control strategy to amend the observed risk assessment, initial risk assessment, to have a proper design space and risk controls strategy to convert all those red and yellow areas into green, or if not all green, then red to yellow, and remaining are green. Nothing should be red remaining here. So it is key thing to handle. So the risk process is when you submit initial risk assessment, you have to also submit to the agency, regulatory agencies in the filing, how did you address it and how satisfied you are. You have to express it in this fashion in a tabular form, and there are some more tabular forms required with quick relevance to the design space and control strategy to give them confidence that you are on right track. QBD applications are there, as I discussed in the morning, on API, as a scope, process development, formulation development, and analytical development as well. And it is expected we should follow it throughout, implement it. But more focus is formulation development, API development is required. API for DMF submission and a formulation for ANDA submission. And this overall, it can be also used in clinical trial, bioequivalence studies. And these data could be correlated with the QBD development process throughout the drug substance, analytical, formulation, manufacturing, bioequivalence, and clinical studies to give a patient a safe, efficacious, and quality product as required in quality target product profile. And this I already described. I'm just recapping few slides here purposely. Now to correlate what we talked just now, we talked earlier what is the expectation of FDA or regulatory agencies. Now we are talking in relation to what we have understood now, the quality target profile, CQA, CPPs together. So from that perspective, impact of quality attributes which gets converted to criticality on the finished product. Any quality attributes has an impact on the finished product becomes CQA. And these critical quality attributes have to be monitored with a right control strategy. This also I discuss. I am again repeating here the format which is to be displayed for the submission of the document. What is important is to write the justification. Now, if you say here, no, identification is not important, you can, but then you have to write here why not. You can write identification is not CQA, but you have to justify why do you say not CQA. Assay is CQA, right here why it is CQA. So that's the way you have to address all the CQAs and non-CQAs. These also I talked to you just now. Deficiencies, another example, API solid state form. The QTPP demonstrate that the CQA and QTPP demonstrate that the form used, API form, is polymorph. Whether the API used is having any polymorph, and that API or a drug substance, if it gets converted to another form or not during the shelf life, what happens to the potency of the drug or the product, and whether patient is affected because of that converted polymorph. So which polymorph you should use for the product manufacturing. So there are several QBD opportunities. It increases the process efficiency, and of course it reduces the cost, and by way of minimizing the rejections and reworking and reprocessing of the steps or a product, increases the level of assurance to a higher level, and yet that increases the quality also, a better quality. There is a transparency in our activity in the development and manufacturing and testing the product, because of the design of experiments and design space, you are moving towards predictable quality of the product. Remember this, don't forget, this is critical. Predictable quality attribute through design of experiments at certain critical process parameters. If you feel that this parameter risk is not being handled, monitored well, then quality will be in question. At that stage itself, you will know it. Then you don't have to proceed further. You can reduce the losses. Compliance issues are reduced. You are streamlining your manufacturing changes, regulatory processes as well, and continuous improvement, which we discuss at length. Patient safety and product efficacy are focused throughout this QBD development and implementing it in manufacturing and testing. Design space and process development are correlated very well, and advantage is your design space, within the design space, your normal operating range is flexible, and you don't have to take regulatory permission within the design space to implement. But if you feel outside the design space, you have to go out for some major advantages, you will have to take regulatory permission. Without that, you cannot change. Remember. And then you are moving towards robust product and process development, and your manufacturing team and your quality assurance is also extremely happy because you are giving robust processes for manufacturing and testing and monitoring at different critical parameters online. And then automatically you are having transparent operations, right first time, CS to CS. You are achieving right first time, remember. You are moving towards right first time. There are as good as nil error, and you are giving, since you are giving robust process and your product development is capabilities and flexibility is improved, you are moving towards zero defect manufacturing the product. Remember, right first time, achieving, you are moving towards zero defect product for better productivity and the process predictability as well. And obviously the regulatory agencies will be very satisfied, customers will be satisfied, and the credibility of organization always goes up, and people will always demand, the doctors, the patients and the hospitals will demand your product more and more. Your business also improves. But how do we achieve online? We talked of online and really real testing, release testing, online testing, inline testing. There are so many terms people have coined for this. Is real-time release testing, RTRT, real-time release testing, with the help of PAT, which is called process analytical technology. Process analytical technology. It has mainly two functions. Mainly two functions is it monitors the process. It analyzes the process. Remember this. So this technology has two main functions, is to monitor the process and to analyze the process. So to analyze means to control the process in such a way to get the real-time release of that particular step. Now look here, the example on your screen. What are different definitions of this? Inline means your probe of your PAT technique or analyzer is inside the container. Nowadays, the probes are untouched to the product and they sense the reaction and send the messages to the machine. So this is inline. Online is the sample is drawn out, analyzed and reported the data. Or at line is drawn out from the system and analyzed next to the production hall, which is called at line. And offline is sending the sample to the quality control lab. Remember these definitions: inline, online, at line, and offline. Here is a FDA's example of real-time release testing at different stages in manufacturing. Here it is a tablet manufacturing, dispensing, blending, sifting, roller compaction, tablet compression, and the pan coating. Now at different stages, different techniques of PAT are used. Mainly, now here you see particle size checking. Here it is a blend uniformity checking. This is a laser diffraction. Even during manufacturing itself, again, there is a particle size estimation required because you are also having lubrication at certain stages. And again, there are sifters used for sifting the material. So at definite particle size you need. And then you are identifying and doing assay of the compressed material. So everything is during manufacturing itself fully automated, and the data you can push it onto your SAP, connect it to the level 4 of your software system or at the site, or you can keep the separate server for this kind of data to collate and study, and here you can release the product for the next step. You don't have to do the sampling at all. You are saving lot of time of sampling You are saving lot of time of documentation. You are saving lot of time of analyzing itself because of NIR analyzer technology as a PAT technology. And as a legal process, as a regulatory legal requirement of every country, final product has to be tested based on agreed specification. So here you save tremendous cost in manufacturing. Yes, while having so many advantages in quality by design, there are challenges as well in development, in manufacturing, and in testing, in assuring the quality. And major change in the organization, in even today, several organizations challenge is a cultural change. Quality culture, the culture of quality has to be imbibed in the organization of every employee. And it is a top-down process, not down to upward. The CEO or managing director of the company is completely responsible for quality culture of the organization. Everybody should think of quality and quality and quality and patient and patient and patient, nothing else while taking any decision in the organization. So this is a must required irrespective of the cost and the losses. By some means, if some batches are failing, you are supposed to reject it. You cannot release substandard batches to the market. Knowledge management is another area which needs to be focused by the organization. The right information, right knowledge, expertise is required. Some SMEs are required in certain areas, depending on the different dosage forms required to the market. In training the people also is a big challenge nowadays, right? Training people. There is a concept of training the trainer as well. I have spent a lot of time with multinationals also in last 15 years after my retirement. Lot of trainings I have done in multinational and generic companies. So right training, right content, right verification of the training is essential. Another challenge is the pressure and the speed of the batches to go to the market from marketing, and the owner or the MD of the company has to be dealt with in a right spirit and right manner without compromising quality. Understanding the risk of failure and they're having right process in place, right identification of the critical quality attributes and critical process parameters is also a challenge, and the right control strategy has to be in place. Another challenge is several people have in mind, I am manufacturing this product for several years. So some batches are rejected, some batches are gone to the market. So what happens? Nothing to worry about. No, that is not the right approach. Airlines, trains, or several travel vehicles, all safety measures are taken, but there are accidents. So we want to avoid any accidents which can create a substandard product and has an impact on the patient. We don't want that to happen. So we have to keep this thing in mind, not to compromise. So avoid this resistance, convince them, do counseling of such people. It is essential. And cost, you don't have to worry about cost. When you think of patient, when you think of the long-term business. And as such, quality by design reduces your cost. PAT implementation, of course, is going to be a challenge because of knowledge of the equipment and the technique to be used, right technique to be used, and you need right experts for training in-house. and these kind of skills which are required by the organization is also very, very important. So how do we move forward? Is fundamental shift of thinking, is designing the right product, right quality, right process, understanding the process, right operation, aim for operational excellence in manufacturing, aim for Six Sigma compliances internally. Your processes should be as good as Six Sigma. Why? These I already discussed with you various guidelines. Now, when we go back to Six Sigma, when we talk of Six Sigma, naturally some people may have a thinking, what is current scenario? Let me tell you very frankly, the current is we are around 3 Sigma, the manufacturing, the process, and we have to enhance it to 6 Sigma. That will happen when we will comply with fully QBD compliances and operational excellence together. That will help to minimize the quality problem and almost zero defect manufacturing, which will have a very positive impact on the business. I'm sure you are aware of one simple example with non-technical, non-pharmacopoeial example is Mumbai Dabbawalas. Millions of tiffins are given every day in Mumbai. In every office, every individual, these tiffins are reaching from their family home towards the offices at right time without mixing each other's tiffins. It is nothing but achieving Six Sigma. Remember this. So we have to achieve Six Sigma. These Mumbai Dabbawalas, they haven't done quality by design. They haven't done any pharmacopoeial manufacturing processes or understanding of that. But simply because of the code word language, they have achieved these par excellence Six Sigma compliances. You can visit their website, Mumbai Dabbawala, and you see this code word. They have the coding, they have the understanding of the coding, which is just par excellence. So we are having paradigm shift from blind compliance to science and risk-based compliance. With the help of quality by design, we are going to a desired state of compliance, and here we talk of continuous improvement to achieve the Six Sigma compliances from 3 Sigma to continuous improvement required with the help of automations to achieve the Six Sigma compliances. To ensure the cost of manufacturing goes down at a achieving desired state. When you achieve QBD, fully realizing the implementation, the cost goes down, and as you go ahead improvement, the cost further goes down. There are several emerging technologies which are in play for last around 5 years. And few of them I'm listing here as emerging technologies, and there is a US FDA document on that advanced emerging technology applications in pharmaceutical innovation and modernization guidance for the industry, which has several technologies, and I'm picking up here few for your discussion with you, is continuous manufacturing, 3D printing, and artificial intelligence, how it can be used for pharmaceutical manufacturing. When we are moving now further after quality by design, we would like to achieve continuous manufacturing process in the staged manner, in a phased manner beyond 2020. Now, some organization has already started in last few years from batch manufacturing to continuous manufacturing to save the time, to give the accuracy of the data, to give the high quality of the product. to minimize the stability problem, to increase the shelf life of the product. And this technology, I was extremely happy to have met Dr. Alan Myerson, inventor of this technology from Massachusetts Institute of Technology, USA, who developed this technology for Novartis, first in the world. And he has handed over everything to Novartis, whole technology. And I was lucky, he gave me almost 3 hours at my request to give me open information on this subject of continuous manufacturing and this technology. And I am highly obliged by Dr. Alan Myerson. And even today we are in continuous touch with each other on this subject. This is called blue sky vision. We are always buying the active drug substance from the manufacturer. We are manufacturing the product, different stages in our plant. What happens that time? You are approving the drug supplier, active drug supplier. You are doing contract with them. They are supplying to you. It takes few days to reach to your plant. After receiving in the factory, it goes to the warehouse. We clean it, the containers. Whenever there is an opportunity, it is sampled in the right area. It is sent to the quality control lab for testing. Quality control lab, whenever gets time to test, they test it, send back the report, and then only the product, active drug, is used for manufacturing. You see how much time lapse is there. It's a huge time gap as well. Lot of time is spent on testing and documentation. Lot of space is used in the warehouse to keep these active drug substances, and that also under controlled temperatures required by that active drug. Now here what we do in this technology, we are only buying intermediates to manufacture active drug substance inside this ultra reactor. Please note my words. We buy intermediates and manufacture active drug substance inside ultra reactors here by using minimum organic solvent, and as soon as active drug is manufactured, it is converted to the tablet immediately without wasting time. And then tablets go for packaging in the bottle or for coating if required, and then goes for packaging. Do you see the advantage here? Tremendous. What do you achieve here? Ultra lean manufacturing. You are getting more improved stable product, minimum impurity level, because you are using the low level of organic solvents to manufacture active drug inside these ultra reactors. And immediately it is consumed to manufacture the tablet. And all these critical processes inside this parameter are monitored with the help of PAT. Process analytical technology is releasing the activity, every critical process inside based on real-time release testing. And that's a tremendous advantage. And I was very, very happy and obliged to Dr. Alan Myerson for explaining this whole technology to me. So the PAT improves the productivity, improves the quality, lowers the risk of failure, increase the sales, obviously. So I always tell my finance guys, I used to tell in the past, 15 years ago, when I was in employment global head, that give me these capexes, and because when I implement PAT, it is profit after tax, not only process analytical technology. It is profit after tax to the organization. It improves my product tremendously. There is heavy productivity. And when you look at the, after complying to this, when you look at the Pharma 4, the industrialization 4.0, from automation perspective, there are several areas or fields which play with each other, machines and equipments and computers together for end-to-end integration for a predictive quality, and which is the key to achieve in the pharmaceutical area, which is technology 4.0, GMP 4.0, quality 4.0, and in the future, all these areas of continuous manufacturing. There is another area which I am going to discuss now is 3D printing, process analytical technology and artificial intelligence together with the help of robotics, cloud computing, data analytics and blockchain are the future challenges to our industry to comply. Let's look at the second interesting technology of 3D printing. And this 3D printing, the first product patented was Aprecia Pharmaceutical Company's product, where API use was levetiracetam. And in 2017, GSK did another product there for their tablets for Parkinson's disease. And this 3D printing technology has the ability to transform whole pharmaceutical industry, and I will explain you why. Let's see this video before we discuss this further. And if you are not able to listen, let me know.
It's a pill you can make with your printer. Yep. A 3D printer to be exact, and it just got approved by the FDA. Trace Gallagher, live with more on that. How do they do that, Trace?
Gretchen, the new drug is called Spritam, and it controls seizures brought on by epilepsy, but it's not the drug that's really revolutionary here. It's the way they're making it, as you say. The company Aprecia Pharmaceuticals using a 3D printer, which is normally used to make things like plastic toys, models, robotic parts. 3D printers work by making things one layer at a time, and printing the drugs allows the layers to be packaged more tightly. So a pill that would normally hold maybe 200 milligrams can now be layered to hold 1,000 milligrams, and yet it dissolves in water as fast as normal pills. The reason this is important is because it allows companies to design drugs for specific patients. In other words, Gretchen Carlson needs an anti-seizure drug, but she needs it with this and with this. Well, that would normally be impossible because the drugs are all mass produced in a factory, but soon your local pharmacy could have a 3D printer to make whatever you want. 3D printers are already being used to make artificial limbs, and dentists use them to make replica jaws, teeth, and dental implants. Instead of using ink in the printer, they use plastic. And experts say they can design virtually anything. It wasn't long ago these 3D printers were hundreds of thousands of dollars, so they were only used to make robotic parts for companies like General Electric and government agencies like NASA. Well, now the price is coming way down, and the printers are affordable for all kinds of small companies. So maybe they print your medicine today, and someday, Gretchen, you walk into a car dealership, you say, I want that car, and they just print it up for you. That's kind of where the technology is going.
Fascinating. All right, but right now it's a pill.
So you see the advantage, the technology of 3D printing. Look at the tablets which are there in the market. Look at the combination. One of the biggest advantage of this is incompatible ingredients can be printed in one tablet without coming in contact. Look at this one example here, ramipril, atenolol, pravastatin, and that also extended release. These are all extended release compartments. Look at different layers of different drugs and how it is used. And it's so advanced technology, and this kind of products are also can be customized. For example, children don't like to take any tablet capsules. But if you give them the drug in the form of their characters they like, Disney characters or other characters they like, in this kind of shapes, they enjoy it. They will enjoy it. They won't realize that they are taking drug. And this is happening in US. So these are customized product for a specific patient. So you need not give whole batch outside. Even pharmacies can manufacture, and when the regulators are approving such activities in USA. When we talk of future, look at that digitally enabled laboratories, automated laboratories, and distributed quality control. Now here it is minimum human intervention. This is another challenge is going to be there. This data was published by McKinsey and Company, and which I have picked up just to share with you how it is going to look like in another 5 years. Whole industry is going to get revolutionized. So you are at a student age, try to pick up this information. So when you enter the industry in next few years, you will start listening a lot of these languages, these words in the industry. So telemedicine, AI, IoT, supply chain, blockchain, these are going to be common words, buzzwords, and throughout you will find the artificial intelligence is going to play a major role. Let me tell you very frankly, it is going to play a major role in the industry, in the product development, as well as in the manufacturing, in testing, and in release as well. And also I would like to share with you one interesting example of training, how this artificial intelligence is used in the training part. I'll just show you one example. Listen to this example video where the trainer is in Las Vegas and teaching Japanese audience in Japanese language. An interesting part, the trainer doesn't know the Japanese language. So how it happens and how this AI is helping us tomorrow, machine learning. See this example. We'll discuss this little more after.
Now, it's a pleasure to be here in Las Vegas to present to you. Now, I get invited to do keynotes across the globe. And while it's easy for me to be here in Las Vegas, it isn't always easy for me to travel across the world. And even when I do, I can't always speak the local language. Well, what if neither language nor distance mattered for me to deliver a fantastic keynote? What if technology could help me be anywhere I needed to be and speak any language I wanted? Well, it can. We are bringing together the power of mixed reality and Azure AI services to create a truly game-changing experience. What you're about to see is an exact hologram of me wearing the same outfit that we recently captured at a mixed reality studio. And I don't speak Japanese, but what if I wanted to deliver my keynote in Japanese? Using Azure AI technology, I can translate my English into Japanese and train it to sound exactly like me. The same voice tones, those same inflections. Now we brought this together, my hologram and Azure AI, to show you what's possible. So first, I'm gonna put on my HoloLens 2 here, and then we'll flip in the room to the special camera so you can see exactly what I'm seeing. Let's get started. First, let me introduce you to Mini-Me. There she is, my perfect holograph. And thanks to the power of HoloLens 2, she just floats right with me. I'm literally holding my hologram, so natural. Now she's a little small to do a keynote. So let's get her up so she can do full-size Japanese keynote. Render keynote.
本当に驚くべきことがあります。私たちは最新の複合現実キャプチャ技術を使用し、私のホログラムを作成しています。実在の人物をホログラムとして見たことがあるかもしれませんが、私が実際に日本語を話しているのが新しいのです。私は日本語を話しませんが、私の声とホログラムは完璧な日本語で話しています。これはニューラルテキスト読み上げと呼ばれる最新の人工知能技術、いわゆるニューラル TTS を使用しています。私たちは自分の声の録音を使用し、私のように聞こえる私自身の個人的な音声署名を作成します。日本語からフランス語、ポルトガル語まで話せます。 今、この技術は私たちが働き、遊び、生活する方法に関して、世界中の国境や障壁を取り除くことができると想像してみてください。まさに SF が現実になるところです。
As you can see, this is mind-blowing technology. And what you just saw was my life-size hologram, my exact replica, rendered here in real time, speaking Japanese with my unique voice signature. To do this, we use mixed reality technology to create my hologram and render it here live. Then we use Azure's speech-to-text capability in English transcription to get my speech. Then use Azure Translate to get the speech into Japanese. And finally, applied neural text-to-speech technology so it sounded exactly like me, just speaking Japanese. And the most amazing part, all of these technologies exist today. The future is here.
So, did you enjoy the video? Great. So, you look at what is happening now in the world from that perspective. This was just a small example, and I assure you definitely in next few years time, this pharma industry is going to revolutionize completely. And just a small suggestion, if possible, try to side by side understand this whole new technology world in relations with the pharmaceutical industry. That will help you a lot as you enter the industry. So why adopt then this technology? Of course, for robust processes, to minimize the wastage, to minimize the reprocessing, reworking, and to minimize the rejections in the market, to satisfy the patients, risk to the patients, reduce the risk to the patient, improve their benefits. Of course, you are going to reduce the cost for sure and reduce the variability. We discussed a lot these variables, and the variability, which is a big issue in the manufacturing and testing, has to be minimized. It helps in shortening the launch time of the product, because your experiments, you are running 2 or 3 factors at a time in your design of experiments, which is helping you to address the optimization compatibility of the parameters issues and get the maximum understanding of the process and the product. And automatically we improve the quality of the product when and the credibility of the organization, image of the organization, when you move towards the higher technology. So with this, I end my presentation.
Thank you very much, sir, for this very informative and a wonderful lecture. All the participants, even many, have posted that the lecture was very wonderful and very informative. So let's go to the session of question and answers.
Sir, one question is here. You can see on question answers, if you can. Q&A.
Yes. Comment on scalability and production capabilities of 3D printing technology. Those are the same way as a normal pharmaceutical products, processes. The scalability is not at all an issue there, and it has a tremendous benefit. Here, nowadays, more focus is on incompatible drugs together, having different layers of those incompatible drugs and customized for specific patients by the pharmacist. So concept of using that today is a totally different aspect. Now, scalability is not an issue because these machines are totally differently designed for this process. You have seen that video where the drug is coming from a hopper slowly falling onto the layer, one layer on the other, one layer on the other. And this speed of manufacturing is also very high. That also can be adjusted based on the type of product, type of ingredient, and the batch size. So scalability, there is no issue. Important is here, the active drugs are falling above each other. And in normal process, they are mixed together. You see there, the scalability issues are more. Here, the tablet is getting constructed itself like a home. If you might have seen some videos or some technology where 3D home construction is there, one after the other layer, machine continuously moves in a planned manner to manufacture a room, to manufacture a dome, to manufacture the toilet, to manufacture the shade. So the same way, this technology, individual drug is falling down from the different hoppers, the layer after the layer, which are coated particles. So the scalability problem is not at all in this, because there is no question of having blend of homogeneity perspective of mixing the 2 or 3 ingredients here.
Sir, one question from my side.
Yes.
I do take the sessions of M.Pharm students for Six Sigma, both
yes.
mathematical and constitutional and the
yes.
philosophical, all 3 components I do take.
Yes.
Now, you were saying that in India, with Indian context, pharmaceutical companies have reached a 3 Sigma level. So the wastage is more than 10%, nearly 10%, more than 10%. Now, automobile companies, in contrast, in India, most of the automobile companies have achieved 6 Sigma. So what is inhibiting us in getting 6 Sigma? Whether it is more important for us than automobiles?
I understand. It's a good question, Dr. Manan. See, it's a mentality. It is a mentality. Now, when engineering companies, auto companies can achieve, why not pharmaceutical? Now, some of the packaging material suppliers, for example, capsules, they do not guarantee 100% defect-free supplies. In such cases, what capsule manufacturers should do? They give in writing, we can give you assurance of 98%, but not 100%. So 2% there is a gap. Look at the compromises happening. Same thing happens from the active drug supplier. Some or the other problem happens. Same thing happens from the excipient suppliers. Same happens from the vial suppliers, rubber plug suppliers. Everybody does some or the other compromises for their own cost benefit without informing the end user. Rejected batches get mixed of the packaging material in the next 10 batches of supplies. So these are the real practical problems, but it is not at all difficult to implement 6 Sigma level in pharmaceutical manufacturing, because we are not ready to accept a 0 defect product yet. And we have a mindset, no, we have so many problems outside. We have so many problems inside. There are suddenly the air conditioning fails. Suddenly differential pressures fail. HEPA filter is not working. The batch is going on. What to do? Packaging is going on. Suddenly the foil supplier has a defect in the foil. Now, no company can check every inch of the foil. They have to depend on the supplier. Initially, when the samples come of the foil, 1 meter, 2 meter or 5 meter is checked, but not in between. So if the uneven thickness of the foil is there, it is going to create some tablets or some capsules, some problem. So we are dependent on so many outside areas to achieve Six Sigma, but it is possible. Now, there is a company in US which supplies the capsules. They say we are giving you capsules at a higher cost, but 100% defect free. Now, looking at the costing in India of the product, competitions and the government controls on the pricing, whether you can match or not is a big question mark. But the companies who are marketing the product to USA, they are importing those capsules in India, using those capsules for marketing in US product, in USA, but not in India. You see the difference. These are practical issues also to the industry. But I appreciate your question, and we should aim to achieve that for sure.
Because Quality Council of India is spending a huge sum of money in implementing Sigma cultures, and 3 Sigma it is, whether it is not even an appreciable situation. At least 5 Sigma, okay. Anyways, it's nice explanation, sir. That is really, it may happen because our ecosystem of pharmaceutical is not as regulated as pharma itself is regulated. So that, yes, these are the issues. Any questions, ma'am?
Yes, sir. I have one question to Ranjit, sir.
Yes.
As sir has rightly said, there are the tools like PAT, DOE, and quality risk management. But I have a particular question with the design expert or design analysis, particularly which software the FDA and other agencies, regulatory agencies, are recommended for the product development, particularly for analysis purposes, sir.
Okay. Now, it is important to understand no regulatory agency in the world recommend any equipment for any purpose. They do not support the equipment manufacturers or software manufacturers because they are regulatory agencies. They are not the business person. They don't look at the business perspective because it will be biased communication. So they don't want that industry to get an impression that we are marketing somebody's product. So they don't get involved into that at all. So they don't recommend anything. Yeah.
Okay, sir. Thank you.
I don't think so. Any questions further from audience side?
So thank you very much, doctor, for inviting me to both the sessions. It was as good as whole day, 4 hours discussion. And I appreciate the initiatives and sincerity of your institute. You as an individual, as a principal of the institute, taking such a great care for your students, enhancing their knowledge, your faculty development program, supporting them from this perspective, having latest information, latest technologies the industry uses. So it's always my pleasure to support such institutions where there is a seriousness from education perspective. So I appreciate and thank you very much for inviting me today for that.
Thank you, sir, for encouraging words. And really, we appreciate you for sparing time for us and other students. Really, because in academics it is very difficult to get the insights from the industries, and with you, really, it was a blessing. So thank you, sir. We are going to conclude this session. Please, ma'am.
Thank you very much, sir. So with this, we complete the day 1 of Charuset Endowment Chair at RPCP. And tomorrow we have session starting at 1:30 p.m. So I request all the participants to join before 1:30. Thank you.
Okay, thank you. Bye.
Thank you. Bye, sir.
Thank you, sir.